Hi, thanks for checking out my talk. My name is Rachel Melamed, and this presentation is about discovering drug effects on disease by bringing together genetic data and epidemiological data across hundreds of common drugs and common diseases. So, one of the big goals of our lab is to discover hidden effects of common drugs on diseases using data. So in order to see if a particular drug affects a particular disease, there are a number of different resources we could use. This includes health data that can show us whether people who take a drug have a changed risk of disease. So this includes uses of drugs to treat a disease or side effects of a drug on a disease. So discovering this from human data is the topic of a preprint that's currently under review, if you want to check that out. But in addition to this human level data, we could instead look at the molecular data. So maybe some connection between the genes affected by the drug and the genes involved in the disease could tell us about drug effects. And this is also the topic of a presentation my student Panos is giving in the same session, which I hope you can check out. But in this project, we wanted to see, can we integrate both of these levels, both human level and genetic data, in one model to learn about new drug effects and to uncover the biology that explains drug effects on phenotypes. So here we propose that drug effects should be predictable based on some connection between the physiological effects of drugs, meaning the genes and pathways altered by the drugs, and the gene networks involved in the phenotype. So we can use data that exists to profile a drug in terms of what cellular processes are affected by that drug. And similarly, we can use GWAS to learn about the causal genes driving a phenotype. And we have, in addition to these two sources, we have that human level data I mentioned, such as the known uses of a medication to treat a phenotype or drug side effects. So bringing all these together, we propose that we can predict and explain drug effects on phenotypes by learning the connections between the processes affected by a drug and genes involved in disease in the form of a weighted network. So how can we learn this weighted network? So we propose that the simplest way to do this could be a linear model. So we translate everything into this linear model. So using EPA ToxCast data, we have measured effects of drugs on diverse cellular endpoints. So we can represent each drug with this vector representing its endpoint effect on diverse cellular processes. And similarly, for each phenotype, we can represent it using GWAS gene-based scores that allows us to make a vector representing any phenotype by its association with different genes that cause that phenotype. So then our model boils down to learning this network W that connects the drug endpoints to the disease genes. And the idea is that we could then do a simple matrix multiplication with this learned network W to get the probability of drug phenotype connection. And we train this W using known drug side effects or indications from the CIDR database. And what's important here is we keep the same W that's trained across 70,000 pairs of drugs and phenotypes. So we call this model DraftNet, the drug and phenotype network, where we learn this W using sparse regularized regression to learn this network connecting drugs to phenotypes. So how do we evaluate this model? We can evaluate our model, by seeing how well we're able to predict effects of drugs. But what we mean by drug effects could mean multiple things. So first, we can predict drug effects on phenotypes, which is what the model was trained to do. So pretty much, as I said, we multiply through to get our predicted drug effect on any phenotype. And then, as with any prediction, we can take our predicted probability of a drug affecting any phenotype and compare that to held out drug effects on phenotypes. And we're able to show very good predictive ability. So you can read more on the preprint or come visit me at the poster. But what I think is really the exciting power of this approach is the ability to predict drug effects on genes that are associated with phenotypes, so the drug disease gene effects. So just to make that more specific, once we have our W, we can multiply through any drug's effect on endpoints to the connection of those endpoints with disease genes to propagate the drug effects to its impact on the disease genome. So that gives us what we call the drug disease genome mapping, which is for all drugs, we estimate each drug's effect on all the disease genes that could have explained the effects of the drugs on the phenotypes. So how do we know that this drug disease genome mapping is actually learning something related to drug biology? So what we do is we compare these mappings with other information about the drugs, information that was not used to train the model. So for example, for any of these disease genes, we can take the drugs that are predicted to impact those disease genes and compare that to drug known target information from drug bank that was not used to train the model and look at the overlap. And by looking at this overlap, we find that drug sharing targets are enriched for drug sharing a disease gene compared to some null model. Of course, the details are in the preprint. So we're able to really support that our mappings reflect known biology of drug effect. So what can we do with this model? So one application beyond predicting phenotype effects is to understand this biology of shared effects of drugs. An interesting example is that we can group together these three drugs that are not entirely obviously related, but we find that they have shared predicted influence on disease genes, including on this gene, CETP, this cholesterol ester transferase protein, which is involved in lipid and cholesterol synthesis. So among these drugs, fenofibrate is an actual cholesterol drug. Triclosan, on the other hand, is an antimicrobial, but it's actually known to act through its effects on fatty acid synthesis. And the third drug in this little group is flutamide, an androgen agonist, but it also impacts dyslipidemia. So it is plausible that all of these drugs could have a shared effect on cholesterol metabolism. So this is just an example, but we have a whole network that we create connecting drugs to each other based on their shared effects on disease genes and connecting drugs to disease genes as well. So we're able to kind of learn a new categorization of drugs. So in conclusion, DraftNet connects drugs to disease genes to propose the biology of drug effect on disease. And we can also use this to predict new effects on held out drugs. So I'd like to thank the students who are involved. Please check out the preprint if you're interested. And also letting you know that we're hiring, and Lowell's an exciting place to live. So please feel free to get in touch. Thank you.